The overall goal of this method is to perform genetic manipulations in the developing chicken auditory organ from embryonic day 4 onward. This is significant because of the difficulty of working with the advanced developmental stage of the chicken embryo.
The main advantage of this technique is that it specifically targets the developing auditory organ in ovo with relative ease compared to other techniques which require complex in utero surgeries.
This method can help answer key questions of auditory sensory development, including how the spatial and temporal pattern of sensory progenitor cell proliferation and hair cell differentiation are coordinated.
To begin this procedure, attach the glass capillary needle using the tubing connected to the glass syringe, which is placed onto the right-hand micromanipulator, and place the electrodes onto the left-hand micromanipulator. Under the microscope, cut the needle tip using a pair of forceps. Next, place 2.5 microliters of plasmid DNA tinted with 0.1% fast green into a dish. Fill the needle with plasmid DNA using the plunger of the syringe attached to the right-hand micromanipulator. Place an egg on its side in a holding device. Then, cut a round window on top of the egg using scissors. After that, place the egg and the holding device under the microscope. Carefully open the two membranes overlaying the embryo using a pair of forceps to expose the right otic vesicle. While stabilizing the head of the embryo with forceps using the left hand, deliver approximately 0.5 microliters of